Welcome, future doctors. Okay, so I want you to think about the number 301. Why? Because between 2008 and 2017 alone, exactly 301 completely new drugs were approved for clinical use. That's a massive number, and honestly, it perfectly highlights why just memorizing a list of drug names is never going to cut it. To actually be a safe, effective physician, you have to master the underlying mechanisms of pharmacology. It's absolutely critical. You aren't just memorizing facts to pass a test here, you are literally learning the language of modern therapeutics.
So let's just jump right in. Welcome to this comprehensive explainer, our masterclass on what is pharmacology. I'll be your guide today, and our goal is simple but huge. We're going to build a deep conceptual mastery of the foundations, and I mean the kind of mastery you need not just to crush your university exams, your vivas, and the NEET PG, but to build a permanent, rock-solid clinical framework for your entire medical career. We're going to take our time, we'll be incredibly thorough, and I promise we're going to make these concepts unforgettable.
Here's our quick roadmap for today's session. First, the roots of pharmacology. Then, pharmacokinetics versus pharmacodynamics. Third, measurement and method. And finally, toxicity and drug testing. Let's get started.
Section 1. The roots of pharmacology. Building the foundation.
Okay, let's take a quick step back into history just for a second to see how this massive modern field actually got its start. Because pharmacology didn't just appear out of thin air, right? It had to be built on pre-existing foundations in physiology, pathology, and chemistry. Let's walk through this high-yield timeline, and definitely pay attention here, because examiners absolutely love testing your grasp of medical history. Take a look at 1847. Rudolf Buchheim created the very first Pharmacology Institute, literally in his own house in Estonia. Just marvel at that bold vision for a moment. He did this in 1847, which is over 10 years before Virchow even proposed the cell theory in 1858, and a full 30 years before Pasteur discovered bacteria as a cause of disease. Buchheim was laying the groundwork for a science before the medical world even fully grasped what cells and bacteria were.
And as you'll see outlined in foundational texts like Ring and Dale's, our modern understanding intricately links those very early historical observations directly to the precise cellular and molecular effects that drive actual therapeutic outcomes in your patients. We've come a long way from Buchheim's basement. Now we're visualizing drugs binding to receptors on a microscopic molecular level. Because ultimately, you just can't understand what a drug does to a disease until you truly understand the disease itself at that cellular level.
Moving right along to Section 2, pharmacokinetics versus pharmacodynamics, the golden rule.
All right, I really need you to lean in for this part, because we are diving into the absolute highest yield concept in this entire introduction. It's the fundamental dichotomy of pharmacology. Honestly, if you remember only one single thing from our session today, make it this perfectly balanced rule. Seriously, memorize it like a mantra. Pharmacokinetics is what the body does to the drug, while pharmacodynamics is what the drug does to the body. It's kind of like a seesaw. Let's say you give your patient an oral dose of paracetamol. Right away, their body starts breaking it down in the liver and filtering it through the kidneys. That is pharmacokinetics. But at the exact same time, that paracetamol is traveling up to the brain to inhibit prostaglandin synthesis and bring down their fever. That is pharmacodynamics.
Let's break down pharmacokinetics first. So the formal textbook definition is the branch dedicated to determining the feat of chemical substances administered to a living organism. But practically speaking, what this means is that we are measuring changes in plasma and urine concentration over time. This gives us the framework for understanding exactly where a drug goes once it's inside the body, and exactly how quickly it gets there.
Now, clinically, pharmacokinetics involves a few specific variables. Imagine a patient taking oral amoxicillin. First off, the body has to absorb it. But how much of that pill actually makes it into the systemic circulation? Well, that's bioavailability, and it can be highly variable. Then, the hepatic CYP450 system basically attacks the drug through phase 1 and phase 2 metabolism to make it water-soluble. And finally, the kidneys or bowels cleared out, resulting in the excretion of the drug in urine or feces. As a doctor, you're going to be constantly monitoring these PK variables, especially, and this is a big one for exams, especially in patients with renal or hepatic failure, where these specific processes are impaired.
Okay, let's flip the coin and look at pharmacodynamics. This is all about the mechanism of action. The strict definition here is events consequent on interaction of the drug with its receptor or other primary site of action. Try to actually visualize this. Picture a beta blocker molecule, like metoprolol, floating in the extracellular fluid. It finds a specific beta-1 receptor on the heart. Muscle binds to it perfectly, like a key fitting into a lock, and slows the heart rate down. That action, that specific biological change, is pharmacodynamics.
You're going to want to highlight these receptor-level actions for your Viva exams. Pharmacodynamics is characterized by some really complex mechanisms of action. It usually involves high-affinity binding, which basically means the drug really wants to stick to its specific receptor. It often has slow on and off rates, which is what dictates how long the clinical effect actually lasts. And all of this results in unusual shaped dose-response curves. You absolutely have to understand this dose-response curve for mastering drug efficacy and for determining the therapeutic index. Remember, the therapeutic index is that incredibly exact margin between healing a patient and harming them.
Now, jumping back to pharmacokinetics just for a quick second, I need you to cement this acronym into your memory right now. ADME. That's absorption, distribution, metabolism, and elimination. These 4 things represent the core pillars of pharmacokinetics that completely dictate drug exposure. If an examiner ever asks you what the body does to the drug, your brain should immediately flash ADME. How does it get in? Where does it go? How is it broken down by the liver? And how is it eliminated by the kidneys?
All right, Section 3, Measurement and Method, Becoming Quantitatives. So now that we have a rock-solid grasp on the definitions, how do we actually measure these drug effects in human beings? Because without measurement, pharmacology is literally just guesswork. There's a brilliant quote from the pioneering pharmacologist J.H. Gattam back in 1942, and he said, "A branch of science comes of age when it becomes quantitative." I just love that because it beautifully illustrates that modern pharmacology relies entirely on precise measurement. In modern medicine, we don't just wave our hands and say a drug lowers blood pressure. No, we measure exactly by how many millimeters of mercury, at what specific microgram dose, over exactly how many hours.
Let's look at some concrete clinical examples of non-invasive human testing methods that you absolutely need to know. How are we quantifying drug effects in a living, breathing human? Well, we use advanced tools like fMRI to measure regional blood flow in the brain. This acts as a really clever surrogate to tell us if neuronal activity is increasing or decreasing in response to a specific psychoactive drug. Or, another great example, we use ultrasonography to precisely measure cardiac performance. Think about actively watching an ejection fraction improve right on the screen after administering an inotrope like dobutamine.
But there's a huge, crucial caveat here. Yes, human studies share the exact same scientific principles as animal studies. And sure, they let us take advantage of the much broader response capabilities of a human subject, like, actually being able to ask them how they feel. But you must always remember, as future doctors, that patient safety and ethical issues are absolutely paramount. We can never ever proceed to human trials without rigorous, ironclad preliminary data. History has unfortunately taught us this the hard way with terrible tragedies like thalidomide. Patient safety is always your ultimate mandate.
Which leads us perfectly into Section 4, Toxicity and Drug Testing, Ensuring Safety. Let's take a look at how all of this foundational science actually builds into the rigorous testing required before a drug ever even gets close to a pharmacy shelf. Toxicity testing is an absolutely non-negotiable phase of drug development. Basically, it involves tests carried out on new drugs in animals to identify any potential hazards way before they're administered to humans. And this is not a quick process, guys. It requires long-term administration of the drug in different species, involving regular, meticulous monitoring for any adverse physiological or biochemical changes. Essentially, we are actively hunting for the poison before we ever search for the cure.
So here are the specific steps of preclinical development that you'll definitely be tested on. Step 1 is PK/PD testing. And hey, notice how our foundational concept of ADME reappears right here. We run ADME studies in laboratory animals to directly link the toxicological or pharmacological effects to the actual plasma concentration of the drug. Then, step 2 is chemical and pharmaceutical development. Look, it's one thing to synthesize a tiny microgram of a drug in a controlled lab. It is a completely different ballgame to prove you can do large-scale synthesis, ensure that the drug remains stable sitting on a shelf for 2 years without degrading into something toxic, and formulate it into a pill that a patient can actually swallow.
As we wrap up this explainer, I want to leave you with a final, slightly provocative thought. We started out today by noting that over 300 new drugs were approved in just a single recent decade. Well, by the time you're all attending physicians, there will be hundreds more. So ask yourself, how are you going to apply your deep conceptual mastery of the golden rule, pharmacokinetics versus pharmacodynamics, to confidently prescribe, monitor, and ensure the safety and efficacy of the therapies you give your future patients? Master these roots today, and I promise you, you'll be ready for any drug that comes your way tomorrow. Keep studying hard, keep visualizing those mechanisms, and I will see you in our next masterclass.